It's Sunday morning. Doing this on a Sunday morning. I need fucking Monster. Hi guys, hope you're well. So if you haven't heard already, but I do suppose that you have, the Pfizer vaccine for COVID-19 is now available in select countries, primarily in the UK. They've now released the data from this phase 3 trial that they were undertaking for this new RNA vaccine, which was published in the New England Journal of Medicine. And I just wanted to go through it with you and explain what some of it means. I read research papers every week. It's just a part of my job. And I have been, I read 2 or 3 a week and I have done for the past 8 or 9 years. So I just wanted to kind of run through it pretty quickly. It's going to be quite a lengthy video because I want to explain all the points that I've included. And also because I've highlighted a lot. So this is the study, and then I've gone through and highlighted various things within it. Oh, it's going to load. It's being a pain, but so I've gone through it, highlighted what I think is important, and I want to go through these, and I'll add the paper itself into any uploads that I do so you can check it out for yourself. So I've actually annotated some of these figures just to make it easier to understand, and then gone through each individual bit, and I want to talk about those. So let's get straight into it. Okay, so we're going to go through this and basically by page. So it's going to be quite a long video. I do apologise for that. So if you are interested, obviously stick around. You don't have to watch it. I will probably put up some other graphics and stuff, but you can just listen to what I have to say. But I am going to talk. I've highlighted specific points in the paper that I find interesting and particularly important. And then we're going to basically talk about them. And I'm also going to go through paper design and structure. So the first page, you can see it says backgrounds, methods, results, and conclusions. This is just the abstract. So if you go to journal websites where you can read papers, they'll have a quick abstract whereby researchers can quickly skim the abstract and see if it might be relevant for their research and their project. Particularly important if you're writing literature reviews. So I've been writing a literature review for 6 months. So just to give you an idea of how methodical this process is and how long it takes to get a review published. Mine is still waiting to be published, so after 6 months. So it can take a long time to try and troll through all this information and find what you want. So firstly, let's go into the methods. So I'm just going to go through the things I've highlighted. You feel free to read the whole thing and ask questions if you want to, but simply because of time, otherwise it's going to take me forever to go through it. So, and I wanted to highlight what these points mean. So the first point, it says, the study was multinational and it was a placebo-controlled, observer-blinded trial. So what that basically means is multinational means it took place over different countries, primarily in the United States, but there were some other sites, such as in Germany. Placebo-controlled means the vaccine has a control. So people that didn't receive the vaccine would receive what is called a control, and that control was saline. So that's what they were injected with instead. And then observer blinded means that not only were the patients blinded to whether they got the actual vaccine candidate or the placebo, but the experimenters were themselves, and that's done by a computer program, which I'll talk about in a little bit. And they were randomized in a 1-to-1 ratio, basically. Again, this is done by computing. Sometimes the numbers between placebo and saline group might be a little bit out, and that's because some people might drop out of the study for various reasons. So next, it says that the vaccine is based on a nucleoside modified RNA vaccine. So a nucleoside, if you haven't heard of that term before, it is similar to a nucleotide. And if you haven't heard that before, a nucleotide are the backbone structures which make your DNA and RNA. So in humans, I think in most other organisms, those molecules are adenine, thymine, guanine, and cytosine. And they contain something called a phosphate group, which binds all of these nucleotides together and forms like a backbone, and that causes the big structure that you see in DNA and RNA. A nucleoside is without that phosphate group. So they just cut it off, and they've used that to modify the vaccine here. So the RNA basically encodes for something called a perfusion stabilized membrane anchored SARS-CoV-2 full-length spike protein. So SARS-CoV-2 or SARS-CoV-2 is the full name of the virus. So it's a SARS-related coronavirus, so similar to SARS in 2003. A spike protein, if you haven't heard of that, the name kind of gives it away. It's basically a protrusion on the viruses, which can bind to our own body's cells via cell surface receptor interaction. So on the surface of all of our cells, there are different receptors that drugs and things can bind to. Viruses can basically hijack these to enter cells. So spike proteins are often described as lockpicks. Almost, they pick the lock of these receptors to gain access to the cells in our body. Next, going on to the results. So almost 44,000 participants underwent randomization. A little bit less than all of the participants received injections. So some maybe were considered to not be eligible for the trial. So they were initially scouted and then said, no, we can't do it. Or they were withdrawn, for example, whereby 21,000 or so were given the vaccine and 21,000 were given the placebo. There were 8 cases of COVID-19 with onset at least 7 days after the second dose of this vaccine. It's a 2-dose vaccine, one on day 1 and the second dose after 21 days. There were 162 cases amongst those assigned to the placebo group, suggesting that the vaccine was 95% effective in preventing COVID infections. And that's pretty stark if you use comparison compared to the placebo group. And they also noted that they saw similar vaccine efficacy, so efficacy means how effective it was, across different subgroups, which they defined by age, sex, race, ethnicity, and baseline body mass index, so BMI, and also the presence of coexisting conditions. So this is particularly pertinent for someone who may have another underlying disorder. It could be another viral infection, it could be heart disease, it could be atherosclerosis, things like that, something associated with old age. It seems to be effective in that group too, which is pretty awesome. So they also note the incidence of serious adverse events was low and was similar in the vaccine and placebo groups, suggesting that at least in the short term that there seems to be no real safety issues associated with the virus. In terms of long-term effects, I'm going to start talking about that and what they actually mention in the discussion about what they're going to do. So the purpose of this entire study was to get emergency approval by the Food and Drug Administration in the United States. So they wanted this paper was published designed for them to read it, and also to provide with the negotiations and the meetings I guess they had with them, just to provide all the data. So some of the jargon can be a bit complicated, I guess, if you're not used to reading scientific papers, and there are some points, most of it is fairly okay. Some of the stats aren't fun, so I'm not going to go through the statistics, and some words are more complicated than they are than they seem to be. They're just other words used to describe quite common things. So I want to break those down. So that's just the abstract. So again, this is going to take some time. So let's get straight into the entire paper. Okay, so there is one thing I will note, and I think it's really important. So in the abstract, it says that 43,000 participants were used. That's a lot, okay, for a phase 3 trial. So according to the FDA, the FDA requires a phase 3 clinical trial to have a minimum of 3,000 participants. So in my eyes, I think that's quite small. I don't think that's a good representation of epidemiological research. I think to get a good scout of the entire population, especially for a vaccine, you need as many people as possible. Obviously, this trial was under quite a tight time frame, so getting enough participants in a short period of time isn't easy, but they've done a brilliant job to get the number that they've done. So I'll provide a graphic now just showing you the FDA website talking about these different trials, and so phase 1, phase 2 and phase 3, and how many people they like to see in them minimally before they accept it for approval. But the website also talks about the timeline of these trials, but obviously a really important point to note is that the phase trials of this was sped up rapidly because it was a different type of approach, and they were Pfizer were also pre-approved funding and pre-approved the go-ahead for the trial, because obviously it being a global health emergency. It's very different to other phase 3 trials, for example Alzheimer's trials, arthritis trials and things like that. Because they're not a global emergency, they have pharma companies and trial bodies have to put applications in, which can take a long time. This hurdle was basically pushed over by regulatory authorities to help speed up the process. And I will also point out, looking at trial numbers, it's really important to look at other clinical trials and the sort of numbers that they were putting out. So all clinical trials have to be made publicly available, but clinicaltrials.gov is a good website for you to check clinical trials that are currently ongoing, and also ones that were published and have data available, and you can find, and they provide links with the actual finished data, so you can actually go and read them. But it's also noted that in other phase 3 trials, there's been considerably less participants in them for effective vaccine candidates. So perfect example of that would be recent ones for Ebola. So some trials for Ebola use far less people, and I believe we do now have an effective vaccine for Ebola. There are many other examples way back from when polio was first discovered, and smallpox trials. They were considerably less than what we have today in terms of participants, but we know the polio and smallpox vaccines are arguably the most effective that we have. So interesting points there. So I just want to go through the entire paper, like I said. So I'm just going to go through the highlighted portions. It's otherwise we will literally be here all day, and no one will watch it. Okay, so we're going to go through everything in yellow and green. So I'll also note before we start, I am dyslexic, so I apologise if it takes me a little bit of time to read some words. It's a pain, I appreciate, but it should be fine. Just some of the scientific words can be a bit fucking annoying on first read, or read through. So, right. We previously reported phase 1 safety and immunogenicity results from clinical trials of the vaccine candidate BNT162b2. So the little 4 here is the reference. So in the reference list at the bottom, you can actually go and check their phase 1 safety data of this vaccine and see that it was safe in their first preliminary testing. And that's basically what it says. So their phase 1 testing, it was safe, and they wanted to then go on to the next stages and see if it could actually be effective for COVID. The vaccine itself was modified by 2 proline mutations to lock it in the perfusion conformation. So, what is proline? So, proline is an organic acid. It's something often coined as a proteinogenic amino acid. So, it's not a traditional amino acid in a sense because it doesn't contain an amine group. So NH2 is an amine group. All amino acids usually contain an amine group, but it is considered to be a proteinogenic amino acid. So just that's what proline is. And then the perfusion, not perfusion, prefusion is basically the conformation of the virus whereby it can fuse to cell receptors. So, i.e. this is the structure that is favourable for it to bind to our cells' receptors. So this is particularly important because you want to be producing the spike protein which is closest as possible to the conformation of the spike proteins on the actual virus, which actively help the virus infect cells. That therefore will potentially help further an immunogenicity profile and effectiveness of this vaccine for this particular virus. Okay, so they provided 2, so that little U sign is micro, so 2 30 microgram doses of the vaccine elicited high neutralizing antibody titers and robust antigen specific CD8 positive and Th1 type CD4 positive T cell responses. So an antibody titer is basically how much of the antibody is produced for COVID following the vaccination. And then CD8 positive and CD4 positive are basically, they're called cluster of cell differentiation, so CD. They are cell surface markers for different cell types. Lots of different cell types express different CD markers. They're primarily associated with the immune system. So CD8 positive and CD4 positive cells are usually T lymphocytes, which are important in adaptive immunity and the production of antibodies, which helps you produce like memory to infections, for example. And they show that it's particularly effective at doing those things, the vaccine, which is obviously very important. So this was just the initial stuff that they were looking at. This is not the phase 2, phase 3 trial of this paper. This is their preliminary data that they're talking about in their introduction to give you a background as to why they think it was beneficial to do. So they can see that, look, it produces antibodies for COVID. We therefore think that it will be beneficial to use in a phase 2, phase 3 trial. They also note that it was 50% more effective than human convalescent serum plasma. So convalescent plasma was used from participants who had previously had COVID, and it was provided to people who were particularly at risk, and to potentially provide them with some protection against the virus, because that plasma will still have antibodies in it. So they're showing that this vaccination is considerably more effective than that. So convalescent therapy, I think, was used for approved for emergency use, and the purpose of this paper is to get emergency use in the United States of the vaccine instead. So that's just kind of summarizing that. In addition, the reactogenicity profile, so reactogenicity just refers to the subset of reactions that occurs soon after vaccination. So that can be local muscle ache, it can be systemic chills, fever, things like that. That's what reactogenicity means. Oh, sorry, that's my emails. Just to let you know that that's what that is. It's again a complicated word for an uncomplicated basically definition. The reactogenicity profile of the vaccine represented mainly short-term local, i.e. injection site, and systemic responses, and these findings supported progression of the candidate into phase 3, which is the study which they're looking at. So collection of phase 2-3 data on vaccine immunogenicity and the durability of the immune response to immunization is ongoing. So they emphasize right at the start, before they even look at the data, that this study is still going on. So this is the preliminary stuff that they wanted to get out so they could get it for emergency funding and push it through for that emergency use, for severe cases and when it needs to be used. So and they've openly said, like, look, this is still ongoing. That's really important. You know, that papers are really transparent. They have to be. This paper was published in the New England Journal of Medicine. It's the biggest medical journal in the world for a reason. The review process is meticulous. To get something published in here it's basic. It makes. So for me as an academic, now, I guess, working at a university, publishing in the New England Journal of Medicine makes a career. Like, it's a big deal. Pharma companies publishing in them is fairly common for trials because they're huge and they have the money. But for individual academics, it's a massive thing. So seeing something published in there you can be sure it's of good quality generally. So that's basically that. So they also note key exclusion criteria. So they say key exclusion criteria included a medical history of COVID-19. Obviously, if you've had COVID-19 before, the vaccine probably, they want to know if the vaccine works in people that haven't been infected by it already. We don't know whether people can get reinfected. There's suggestions that you can, but they just want to know, look, if you haven't been exposed to it, can you just be okay and kind of get over it, sort of thing, with the vaccine if you haven't been exposed to it. So they also note that other key exclusion criteria is people that are using immunosuppressive therapy. So this might be someone that has an autoimmune disease, arthritis is a good example, multiple sclerosis. Sometimes you all have immunosuppressive therapies. The reason being is that these are autoimmune disease. What that means is the immune system is attacking the body's own organs. So Crohn's disease, the immune system attacks the gut. Multiple sclerosis, the immune system attacks neurons. Rheumatoid arthritis, the immune system attacks your joints. So this trial, and specifically this study, doesn't look at that. So they talk about that they're going to do that a bit later, but they specifically emphasize that this initial data is not on that. So if you have any of those conditions, that's something to note before rushing out and getting a vaccine. It's best to wait and find out more data, which they will publish soon, specifically on those diseases. And so they also note, finally, in the introduction, that an independent data and safety monitoring board reviewed the efficacy and unblinded safety data. That's obviously really crucial. So the data was independently analysed as well and unblinded. So someone that's not associated with Pfizer or this study looked at the data to make sure it was legit. So let's get into the actual methods and the data itself. Now we're going to go into the trial procedures. So section, which I think is pretty important. So the last two bits I was talking about was actually the methods, so the key exclusion criteria and then also the independent data and safety monitoring board section. So now we're going to look into the trial procedures before going into initial safety. So trial procedures. With the use of an interactive web-based system, Participants in the trial were randomly assigned in a 1-to-1 ratio to receive 30 micrograms of the vaccine, so 0.3 mil volume per dose. It's pretty small volume, which is good, or saline placebo. So saline is just a salt solution, usually sodium chloride, but it can be potassium chloride and calcium chloride. It's usually, it's normally ions, salt ions, which are common in the blood. So calcium, potassium, sodium are all common ones that they include. So site staff who were responsible for safety evaluation and were unaware of group assignments observed participants for 30 minutes after vaccination for any acute reactions. So they were looking for acute allergic reactions primarily. Sometimes allergic reactions can happen later than that. And as you've seen in the news, that there appears to be some allergic reactions in some participants. But again, just because you're allergic to something does not mean it's globally unsafe. Think about, peanuts are very unsafe for those who are allergic to them, but generally in the worldwide population, it's fine. Same, we're allergic to many things, pollen, dust, certain fabrics, certain foods, certain drinks. So just because someone might be allergic to something doesn't mean that they're not beneficial. Think about it. Someone might be allergic to a bee sting, but are you saying that bees aren't beneficial to the planet? So it's something like that as a metaphor, I guess. So. But yeah, the site staff, so these would be the nurses, things like that, also didn't know what vaccine they got, vaccine the participants got, whether it be the placebo or the vaccine itself. So that's really important because that avoids them being biased towards one person or the other based on if they know whether they got the vaccine or not. So blinding everyone to who got what is very, very important for clinical trials. Okay, so we're going to initially go to the initial safety section. So the primary endpoints of this trial were solicited, specific, local or systemic adverse events and use of antipyretic or pain medication within 7 days after the receipt of each dose of vaccine or placebo. So that's the first part that I'm going to talk about. So primary endpoints. So firstly, they were looking at solicited specific local. So again, this would be local inflammation, local pain. You can often get a really strong muscle ache sometimes. Like I got my tetanus shot, I think about 6 weeks ago, maybe 8 weeks ago, and I had local pain for a week. Sometimes that happens. It doesn't happen so much in other people. So my Bronte, my girlfriend, she had more pain than I did, and that's just, it depends on the person. The immune response can be different per person. And then systemic adverse events. So these are things like muscle ache, diarrhea, vomiting, fatigue, fever, systemic events based on the vaccine, usually because its immune response travels in the blood, so it's going to go everywhere. It can go to your muscles and make you feel a bit. Immune response in the muscles can make you feel really achy. It can sometimes feel I guess, similar to 3 days after doing legs at the gym. And the pain isn't quite severe anymore, but you still it still hurts to move a bit. You still need a good massage and stuff like that. So it's a good kind of comparison, I think. So. And then also looking at the use of antipyretic. So this is anti-fever medication. So that's what antipyretic medication is. And then also pain medication. So whether that just say keeping an eye on people that needed those medication. And they kept those analysis for comparison in the different treatment groups and for the graphs. And then this was up to 7 days after receipt of each dose of the vaccine. But that isn't all. They also looked at unsolicited serious adverse events through 6 months after the second dose. However, adverse events data through approximately 14 weeks after the second dose are included in this report. So, really important thing here, okay? So 14 weeks is the reason that it's not an entire 6-month period at the moment is because they wanted to get the data out as quick as possible because there was probably push from governments that they need a vaccine. So so getting data out with acute reports of side effects isn't uncommon, and I will really emphasize that, specifically for vaccines that are required urgently. So I think something might have happened similarly with Ebola, same with polio, smallpox, etc. They do note that there will be providing data for up to 6 months initially, but that's not all. And this is in terms of adverse reactions. So people that are worried about safety and chronic safety or chronic side effects of vaccines, there will be more data coming. I really want to emphasize that. It's not like they're not just reporting it. And I will also do the flip side of the coin. There is suggestions that long-term effects of COVID can cause lung sclerosis, or lung sclerosis means, lung fibrosis, which means scarring, chronic fatigue, and various neurological issues. For example, if you produce neuroinflammation in response to this infection, that can have massive consequences for a range of neurological based disorders. So I'm a neuroscientist by training. I'm primarily interested in neuropharmacological interactions, so what drugs can be used for different neurodegenerative diseases. Suggestions that inflammation is a key determinant in the early progression of a multiple array of diseases, including Alzheimer's, Parkinson's, MS, and a range of others. So, including things like traumatic brain injury and stroke. So, inflammation in the brain might help facilitate a hemorrhage, for example. So, it's some things to keep an eye on, and it's always good to look at two sides of the coin, for example. So, but they're just emphasising that, look, this is initial acute side effects data that they're talking about, and we will include more as time goes on. Again, they're still gathering that data, but they've obviously got 50,000 or so participants that they've studied. They've got great data. It needs to be published. The public need to know about it, and so do the government bodies. Okay, so per protocol, safety results for participants infected with HIV, that was almost 200 people, will be analysed separately and are not included here. So this is quite a common thing to do in statistics. So if there is a population that differs from the rest, it is always good to analyse the data separately. So a good example of this is, so I'm currently working on models of ALS, so amyotrophic lateral sclerosis, often otherwise coined motor neuron disease. It disproportionately affects women. I think around 70% of people that have ALS are women. So when we use animal research for ALS, it's important to analyse female mice differently to males because the disease progression may be different. So pooling the data together may not give you a clear-cut change or effect of drugs or treatments because males may respond differently to females. And that's basically what they're saying here in terms of infections. So you could have another infection such as HIV, and that may result in different responses in those patients. So it might be better to analyse those people individually from the rest of the population. So they only have 200 patients with HIV here, so obviously it's a small population. It might be harder to recruit those participants for various reasons. One, people might not want to admit that they have HIV, and also finding them in the first place that might be signed up to trial registries and things like that. So 200 patients is not a good representation of the entire population of people that have HIV. It's a low number. So getting more data from people that have HIV would definitely be important to increase the power of this independent analysis that they've done. Okay. So we are now going to go on to the efficacy portion, which means just how effective the vaccine is. So the only portion of the efficacy part I'm going to talk about briefly is the small bit I've highlighted, and that is the main safety subset as defined by the FDA with a median of 2 months of follow-up as of October 9th, 2020, consisted of 37,000 or so persons, and the reactogenicity subset consisted of 8,183 persons. So the reactogenicity is the immediate effects. They followed 8,000 people to get those immediate effects, such as what's happening, are there any serious immediate effects that they're seeing, and then the general adverse effects for 2 months follow-up was for basically everybody. So that's important. So the local reactogenicity was only a subset, and longer term side effects or 2 month side effects were everyone. So the one thing I would note is, why wouldn't you just do the reactogenicity subset for everybody? I imagine that there was probably some form of caveat in the way for that, maybe getting access to them, the resources they had, something like that. Normally, so at the end of a paper, they have a discussion section where they talk about, I guess, flaws or things that they can improve on or things that they can expand on. They don't talk about this here, I don't think. They do have supplementary data, which I haven't dug into. But there is probably a reason behind it. I think 8,000 people, again, way exceeds the FDA's required 3,000. But having some more would be, in my opinion, would probably be beneficial to them. I imagine that they probably will include more data because I said they are still providing that. So just something to note.